So, good afternoon. In the first unit, we have seen the nature of chromosome in bacteria, yeast and viruses. Later on, we have seen the DNA replication in detail. The DNA, how does the DNA replicates? The DNA replicates semi-conservatively, and as a evidence, we have seen Michelson and Stahl's experiment. Also, we have seen conservative and dispersive mode of replication. Then we have seen what are the different enzymes involved in DNA replication. Also, we have seen the mechanism of DNA replication, how the DNA replicates, the DNA replication of leading strand or the leading strand synthesis and the lagging strand synthesis. Also, we have seen the characteristics of DNA polymerases, and lastly we have seen the function of DNA. Now we are starting with another unit. In continuation with DNA replication, I am first starting with transcription and translation process. So what is transcription process? Transcription is nothing but whatever the genetic information is stored in the DNA is getting transcribed in a nucleotide chain which is single strand, and that single strand nucleotide chain is nothing but a synthesis of messenger RNA, and this process is called as transcription. Later on, the messenger RNA binds with the ribosomes and several other enzymes and protein molecules with utilization of energy, and the protein synthesis takes place. The synthesis of protein by taking the information encoded in the messenger RNA in the form of a genetic code which constitutes three bases. That particular information which is specified in a genetic code, it joins the specific amino acid in the protein molecule. So, protein molecule is nothing but a polypeptide of amino acids. So, this process of protein synthesis is called as a translation. Okay. So, now first of all we will see the central dogma of molecular biology. Now, what is the central dogma of molecular biology? So, you know that the genetic information is stored in the DNA. So, when DNA undergoes a DNA replication, it makes its own copy, and this genetic information is stored in the particular parent cell. So, when it is undergoing a replication during the that is what is called as binary fission. So, when the two daughter cells are to be formed, so this DNA segregates after synthesis of a new strand, so that one daughter retains the single strand from the parent and new strand is being synthesized, and the other daughter cell also conserves one DNA strand from the parent, and the other strand is a newly synthesized one. Later on, whatever the genetic information is stored, this is transcribed into a RNA. So, this process is called as a transcription process. So, this is called as a transcription process. So, in transcription, messenger RNA is usually found. So, this is the synthesis of messenger RNA. However, in few viruses like HIV virus, you will see that RNA is the genetic material. So, when RNA is the genetic material, then this RNA can itself undergo a replication process in order to maintain its progeny, or in order to undergo multiplication and to form mature virus particles, the RNA has to undergo a replication process. So, this replication process usually is carried out in presence of RNA polymerases. So, RNA polymerase can also undergo a replication process to make its duplicate molecule. Now, however, this RNA, it undergoes a reverse transcription in presence of reverse transcriptase enzyme. So, HIV, it can form a DNA. So, when it is forming a DNA, then it uses the reverse transcriptase enzyme. So, this reverse transcriptase enzyme makes DNA from the RNA molecule. Later on, this RNA is a single strand polynucleotide. Now, what is the difference between DNA and RNA? Usually DNA contains thymine and it is a double stranded structure, but RNA or the messenger RNA is a single strand molecule, and instead of thymine you will have the uracil. Okay, so this is a single stranded and it has uracil instead of thymine. Now, whatever the information is stored in this messenger RNA is used for synthesizing the protein molecule. So, this process is called as a translation process. So, translation means the information present in the nucleotide sequence of RNA, the 3 nucleotides making a genetic code, and this genetic code specifies amino acids, and this amino acid is now making, after formation of peptide linkage, a protein molecule. So, in brief, we will say that the central dogma is the flow of genetic information from DNA to RNA and from RNA to protein. So, this is a well illustrated figure which shows the DNA replication, transcription and translation. So, this is your double stranded DNA, and this double stranded DNA usually when it undergoes a replication process, it makes its duplicate molecule and is segregated in the daughter cells. Then this is your RNA polymerase which makes the messenger RNA. Messenger RNA is usually a positive sense strand. You call it as the messenger RNA, which is a positively sense strand, and this sense strand is mainly used for the translation process. So, this RNA makes the protein molecule when the messenger RNA binds with the ribosome subunits, and this ribosome subunits provide a protein synthetic machinery for formation of the peptide linkage in protein synthesis. However, this sense strand can also undergo a reverse transcription in presence of reverse transcriptase enzyme to form a DNA molecule. So, in HIV you will see that the DNA molecule, why it is forming the DNA molecule? In HIV virus, usually this RNA is the genetic material. It gets converted into the DNA, and after getting converted into the DNA, this DNA fragment or the DNA of that particular HIV virus is getting integrated into the human or animal's chromosome. This, in terms, if it were in the form of a RNA, it could not have got integrated into the human chromosome. So, when it has to undergo integration into the human chromosome or into the DNA of the human chromosome in the nuclei, then it has to be converted into double stranded RNA. So, this reverse transcriptase enzyme makes the positive sense RNA into the DNA molecule. Later on, this is your usually this positive sense RNA is undergoing a replication process to make its duplicate copy. Usually what happens, certain viruses, they have a single stranded RNA molecule as their genetic material. Getting my point? Key viruses, sometimes they have single strand RNA as a genetic material. So, in order to undergo a replication process, that particular strand has to get converted into double stranded RNA molecule. Okay. So, that RNA molecule means, in order to form a particular replicative form, usually the sense strand is undergoing a replication to form a negative sense, and then by means of replication process, that particular one strand is acting as a template to synthesize the another strand by mainly by rolling circle mechanism, and therefore usually this particular sense RNA, negative sense RNA, then becomes the positive sense RNA by means of RNA dependent that is RNA polymerase enzyme. So this is the replication process in few viruses. Now the translation process, that is messenger RNA, which is coding the nucleotide sequence for and specifying a particular type of amino acid, that enzyme is being polynucleotide, forming a polynucleotide chain in the protein molecule. So this is your peptide linkage and this is your amino acid molecules, okay, amino acids. So this is another diagram. Once again it is explaining the same thing, only thing is that the structure of messenger RNA is a different thing. See, the DNA is huge here, okay. So it is containing a double strand and it is made up of many nucleotide sequence. Now on the genomic DNA, this is your double stranded helix genomic DNA, you have segments of or fragments which are considered as gene. So messenger RNA is formed only from one particular segment located on the genome. So say for example this particular region from here till here, so this is the gene region. So this region is now having, this region is having a that is your DNA is acting as a template for the synthesis of messenger RNA, and RNA polymerase is the enzyme which synthesizes the messenger RNA. So you have a promoter region, then you have a transcription from where the transcription starts. This is your RNA coding gene, plus you have the introns which has to be spliced out or cut it out or you remove it out. So this after transcription you will get a polymer that is messenger RNA. Now this messenger RNA usually it has a 5' cap, you have a 3' poly A tail, these are your terminal repeats and these are also your starting terminal repeats. So this has to be removed or spliced out. And also you have from where the usually the transcription starts from start codon. So this is your start codon where the translation begins or it is starting specifying that amino acid. So this is the coding region which specifies the amino acids and forms a protein molecule. So and here you have a stop codon. So this is the structure of messenger RNA we have already seen in detail previously in the first unit. Now usually what happens, the prokaryotic messenger RNA does not undergo any processing. This particular structure is usually observed in the eukaryotic messenger RNA because the eukaryotic messenger RNA is synthesized in the nucleus and protein synthesis takes place in the cytoplasm. But this is not the case in the prokaryotic cell. In prokaryotic cell this is not the case. Why? Because nucleoid is in vicinity with the cytoplasm. Means what? Nucleoid हा cytoplasm मध्येच आहे. त्याच्यामुळे त्याला न्यूक्लियर मेम्ब्रेन नसल्यामुळे न्यूक्लियोइडला न्यूक्लियर मेम्ब्रेन नसल्यामुळे तो ऑलरेडी सायटोप्लाझमच्या टच मध्ये आहे. इट इज इन द विसिनिटी ऑफ द सायटोप्लाझम. नाउ व्हाट हॅपन्स की व्हेन द जीन located on that nucleoid is transcribed and making a messenger RNA, that messenger RNA is directly coming out in the cytoplasm. It binds with the ribosome subunits and starts the protein synthesis. So you can see number of ribosome subunits added to a single molecule of that particular messenger RNA. It means that you will have polyribosome units located on a single messenger RNA, and therefore you will get multiple number of protein molecules from a single messenger RNA. Okay, but in case of eukaryotic messenger RNA, the protein synthesis is something different. Why? Because messenger RNA is synthesized in the nuclei, means the transcription process takes place in the nuclei, and after transcription usually the messenger RNA comes from the nuclear pore. The nuclear membrane is having a nuclear pore, and from the nuclear pore the messenger RNA is getting out, then it is entering into the cytoplasm, and after entering into the cytoplasm it comes and binds with the ribosome subunits and starts the protein molecule. So, in order to come out from the nuclei, it has to be protected from several exonuclease activity, or it has to be get protected from nucleus activity at both the end, and therefore the messenger RNA is having a 5' cap and it is having a poly A tail. Okay. So, when it comes into the cytoplasm, then the protein synthesis takes place. So, after protein formation, means when the translation is over, you will get a protein molecule, and this protein molecule is nothing but a polypeptide chain which is having the amino acid. Now, you have 20 different amino acids which forms a protein molecule. Okay. So, you will see that the 4 nucleotides in the DNA ATGC is getting transcribed into a messenger RNA, that is AUGC, and this AUGC 4 letter code is getting translated into a language of, that is protein, and therefore that is the amino acids in the protein molecule. So, in eukaryotes after the synthesis of protein molecule, usually you will see the post translational modification. You will see post translational modification. How? Some amino acids will get cleaved, there will be a addition of phosphate group, there will be a glycolysis process, means addition of different carbohydrate moieties, then ubiquitation and many more. But this is not related with your syllabus. You will study this in your PG or postgraduate. So, you are leaving this at this right now. Okay. Next, now we are looking for step by step what is the central dogma. First is the DNA replication. Now, I am not going in detail, already we have studied in. So, DNA replication is semi-consecutive. Yes. Huh? Hello? Hello?
काही नाही झालं मॅडम.
Okay. So, DNA replication usually takes place in 3 stages: initiation, elongation and termination. In initiation, helicase unwinds the DNA forming a replication fork. Multiple replication forks are usually found in the eukaryotic DNA, but only a single replication fork in found is that is the 2 replication forks at opposite direction is found in the prokaryotic cell. So, at the origin of replication only the DNA replication starts, and then this is the eukaryotic where you find a multiple replication forks. So, this is one origin of replication, this is another origin of replication. So, you can have multiple number of replication forks in the eukaryotic DNA, eukaryotic system. So, elongation is nothing but the adding new nucleotides. So, RNA primer enzyme synthesize a RNA primer which is usually 10 to 12 nucleotides in sequence, and it is bound to the template DNA in the that is 9 merge sequences. We have seen the 4 9 merge sequences. Later on, the unwinding of the template DNA starts and it forms a open complex, then it forms a pre-initiation complex, and then the polymerization starts with the DNA polymerase 3, which reads the template DNA in 3' to 5' direction and adds new complementary nucleotides in the 5' to 3' direction. So, when it is usually adding the nucleotide in 5' to 3' direction and taking the template strand, that is 3' to 5' strand, then it is called as a leading strand synthesis. But when the another strand of DNA, that is the parent DNA strand, is having 5' to 3' direction, then since the DNA helix is having a anti-parallel nature, because one strand is running from 5' to 3' and the another strand is running from 3' to 5'. As you see that this is your parent DNA 5' to 3' and the another strand is running from, that is 3' to 5'. So, when 3' to 5' strand is usually used as a template, the synthesis occurs in a 5' to 3' and this strand is synthesized in the that is leading way. Okay. However, the second strand, which is opposite, or what we say that it is complementarity to in nature. Therefore, since this template strand is having 5' to 3' and replication always takes place from 5' to 3', it has to move in the opposite direction, and therefore, when this strand synthesis takes place, usually it is taking place discontinuously in this way. So, the replication fork is moving in this direction and your Okazaki fragments or the lagging strand synthesis takes place in this way. Okay. So, this already we have seen. Then we have also seen the DNA replication termination. How the termination occurs in prokaryotes. You have a TER sites to which the TUS protein or terminal utilization substance adds to it, and because of binding of the TUS substance or the TUS proteins, usually the helicase enzyme cannot move forward in the forward direction, and therefore the replication fork will not move and the DNA polymerase will not synthesize the nucleotides. So, when it will not synthesize the nucleotide, means the DNA replication process stops here or gets terminated here. After that, RNA primers are removed and the DNA polymerase 1 undergoes short stretches of this particular DNA. It replaces this particular DNA of RNA primer, and then later on the gaps are filled with the DNA ligase and you will get each new DNA molecule synthesized on the template DNA strand. Now, we will see the transcription process. Now, what is transcription process? Usually, you have two strands of DNA. In a DNA, since it is a double helix structure, you have two strands. But for RNA synthesis, among those two strands, only one strand is acting as a template. This is your first point to be noticed or to be noted. From the two strands of DNA, only one of the strand is acting as a template. Second, second point, instead of DNTPs, you require, that is NTPs only, means uracil, adenine, cytosine and guanine, cytosine and guanine. Only these four are required. In DNA you have thymine, adenine, cytosine and guanine. So, this is the difference. Now, in RNA you have ribose sugar. It is called as a ribonucleic acid. So, in ribose sugar, here you have OH group, but in deoxyribose sugar you have only H group. So, this is the difference between the DNA and the RNA. RNA is very important type of nucleic acid because it plays an important role in the production of protein molecules. Also, RNA is acting as a carrier of instruction from the DNA out of the nucleus to the ribosome subunits in order to synthesize a protein molecule. Similarly, RNA in certain cases they are acting like a enzymes, and therefore they are called as riboenzymes, and therefore these RNAs are responsible for regulating or controlling certain processes, metabolic processes. Okay. So RNA synthesis, usually the genome of any organism contains all the information which makes that particular organism. So the information is encoded in various types of genes. So this is your eukaryotic cell. Eukaryotic cell is having multiple number of genes. The prokaryotic cell is also having many number of genes, but when we compare the genes in the prokaryotic cell, eukaryotic cells are having maximum number of genes. Okay. For example, the prokaryotic cell can have only that is 15,000 that is 1000 to 1500 base in one particular gene. That is, the gene size of a prokaryote is 1000 to 1500 bases, and it can have 4 genes, for example, in a simple prokaryotic cell like viruses. That is, if you observe a small viruses, then you can have only 4 genes. But when you compare with the bacteria, bacteria can have thousands of genes. Bacteria can have thousands of genes, means at least 1000 genes can be located on its nucleoid DNA or the bacterial chromosome. But when you compare with the humans, human has, that is 3.1 billion bases in its bases in that complete genome or the DNA, and it has near about 30,000 to 40,000 genes located on the DNA or the human chromosome. In 23 chromosomes, you will have near about 20,000 genes which are in functional state. In functional form, you have near about 20,000 genes. So this is how, as per the complexity of organism, you will have the number of genes. So this you are going to see in more detail in the second unit. Okay. So now what 4 different types of RNAs are usually found or transcribed from the DNA. So these are the messenger RNA, which encodes the amino acid sequence of a polypeptide. Then you have the tRNA or the transfer RNA, or you call it as the soluble RNA, which brings, what is the function of tRNA? They bring the amino acid to the ribosomes during the translation process or during the protein synthesis. Then ribosomal RNA with ribosomal proteins, they make up the ribosomes, the organelle which translates the messenger RNA. Okay. So this ribosomal RNA is also important for the protein synthesis, and small nuclear RNA or the snRNA, with binding with the protein, it forms complexes, and these complexes are used in RNA processing in the eukaryotics. But small nuclear RNAs, they are not found in the prokaryotic cell. They are usually found in the eukaryotic cell. As well as you have heterogeneous nuclear RNAs, then siRNA, small interfering RNAs, etc., that we have seen earlier. Okay. So now how the RNA synthesis takes place? Now RNA synthesis or the transcription process is usually the synthesis of a messenger RNA. The messenger RNAs actually carries the code which specifies the amino acid sequence in the protein molecule. So making messenger RNA, it starts with the protein encoding gene on a template strand of DNA. So if you have this particular 2 DNA strands, among those 2 DNA strands, only this is acting for as a template for the synthesis of messenger RNA. As I have already mentioned, that two strands of DNA are not acting as a template, only one strand is acting as a template for the synthesis of messenger RNA. Okay. So, this is your promoter, then this is your RNA coding region, and this is your terminator. The complete gene is residing in this particular location. Then RNA synthesis, how it takes place. Again, as we have seen the DNA replication, the RNA synthesis or transcription also takes place in 3 stages or 3 steps. One is the initiation. Now, RNA polymerase, it binds to the promoter region. Okay. So, promoter region is a special nucleotide sequence where the RNA polymerase recognize means odakhne. It recognizes this particular promoter region and binds successfully. Okay? And after that, after successful binding, it signals the beginning of a gene. So, this is the gene where it is going to be transcribed. The RNA polymerase, usually it binds to the Tata box. This is the promoter region where it has a special nucleotide sequence which you call it as a Tata box. And the binding of RNA polymerase to Tata box is brought about by the transcription factors. This we will see in detail in a transcription process. Okay. So, then later on the double helix unwinds and the transcription begins. Then during the elongation process, the second step is the elongation process. In elongation, RNA polymerase moves along the protein encoding gene and adds a RNA nucleotide in 5 prime. See, for the DNA replication also takes place in 5 prime to 3 prime direction, and RNA synthesis also usually takes place, usually means always it takes place in 5 prime to 3 prime direction only. And the RNA polymerase is having a processivity rate, that is up to 60 nucleotides can be added per second. Then termination of RNA synthesis or the transcription is usually brought about at the termination region. So, here you have two, usually in prokaryotes, the termination of transcription is usually brought about by row dependent factor or row independent. So, when you have a row dependent, then usually the row proteins binds to this termination site and it brings about the termination. So, you have many different factors as well as the enzymes which brings about the termination process. But in row independent, usually the complementary sequence is being synthesized in the 3 prime end, which forms a hairpin loop. This we are going to see in detail in the transcription process. So, here just you have to give it as per your central dogma. So, you have to just mention the initiation process, elongation and termination process. So, the product of these 3 steps usually is either a immature RNA or it can be a pre-messenger RNA. Pre-messenger RNA means it is a precursor of messenger RNA. Later on, this immature messenger RNA, immature is of no use, it is fallen off and it is not having any function. But the pre-messenger RNA undergoes a modification, or you call it as transcriptional modification, in which there is addition of 5 prime cap at the 5 prime end and a addition of poly A tail at the 3 prime end. Okay. So, likewise then you have a splicing of introns. So, intervening sequences are spliced off, which is of no use in protein synthesis, and only the region is kept in the messenger RNA which codes for or which specifies particular amino acids in that particular protein molecule. Now, we will see the translation. So, once again, in central dogma, the genetic information is flown from DNA to RNA and from RNA to protein molecule. So you have seen the transcription process. Transcript means what? See, if you are going abroad for study, after completion of your BSc, if you want to do MS or MSc, we call MSc in India, but we call Master of Science MS in abroad, whether it is a UK or whether it is a US. So if you are going there, then that particular university in which you want to seek admission, they demand for a transcript from where you have done your bachelor's degree. So for example, if you are done your BSc from SRT University, then after applying at US or UK for MS, that university will demand a transcript from SRT University. When this transcript is nothing but you have got the mark memos in your hand, okay, but these mark memos are to be verified by the university from US or UK. So that verification of your marks in your mark memo is usually done with the transcript which has acquired from the SRT. So you have to apply it to the, that is, examination section for the transcript, and you will get the transcript within 8 days. So within 8 days you will get the transcript, means the complete BSc marks along with your score is actually transcript is having all the 3 years marks. The mark memo is of a single year which you are having, but the transcript is having the complete marks which you have obtained in all the 3 years. That particular transcript means it is a language. Now we are moving to our, that is, transcription process. So what is transcript? Transcript is nothing but a nucleotide sequence. जसे तुमच्या मार्क मेमो मध्ये मार्क्स आहेत तेच मार्क्स तुमच्या ट्रान्सक्रिप्ट मध्ये मिळतील. पण ट्रान्सक्रिप्ट, मग तुम्हाला वाटेल की मार्क मेमोची गरज काय आहे? तुमची मार्क मेमोज डायरेक्टली त्या US किंवा UK युनिव्हर्सिटीमध्ये ॲक्सेप्ट का होत नाही? नाही होत, कारण त्यांना व्हेरीफाय करून घ्यावी लागते आणि ती परस्पर मागवून घेत असते. ती तुमच्या हातात युजली मिळत नाही, ती सील बंद पॅकेटमध्ये मिळते आणि ती तुम्हाला सबमिट करावी लागते. सो यू कॅनॉट टॅम्पर दॅट पर्टिक्युलर ट्रान्सक्रिप्ट, कारण त्यात पूर्ण तुमचा लेखाजोखा असतो. तुम्हाला किती मार्क्स मिळालेत, काय मिळालेत, म्हणजेच काय की तुमचे जे ॲक्च्युअल मार्क्स तुम्हाला जे मिळाले, तुमच्या मार्क मेमो आहेत तेच मार्क्स तुमच्या त्या युनिव्हर्सिटीला सबमिट होतात. त्याला म्हणतात ट्रान्सक्रिप्ट. आता इथे पण ट्रान्सक्रिप्ट कसं म्हणतोय आपण की DNAच्या ज्या न्यूक्लियोटाइड्स आहेत, जो टेम्पलेट यूज केलेला आहे तो टेम्पलेट जेव्हा मेसेंजर RNA बनवते, त्याच्या ज्या न्यूक्लियोटाइड सिक्वेन्स आहेत त्या न्यूक्लियोटाइड्स जर का आपण घेतल्या तर एक्झॅक्ट कॉपी आहे ती नॉन कोडिंग जीनची. लक्षात आले मी काय म्हणते? समजा जर का तुमचा हा स्ट्रँड मेसेंजर RNA कोड करत असेल, तर मेसेंजर RNA कोड केल्यानंतर मेसेंजर RNAची जी न्यूक्लियोटाइड सिक्वेन्स असेल ती एक्झॅक्ट कॉपी कशाची असेल? या वरच्या स्टँडची एक्झॅक्ट कॉपी असेल. फक्त आणि फक्त काय डिफरन्स असेल की टीच्या ऐवजी मेसेंजर RNA मध्ये U असेल. दॅट्स ऑल. म्हणून त्याला ट्रान्सक्रिप्शन प्रोसेस असं म्हणतात. नाव यू आर मूव्हिंग टू द ट्रान्सलेशन. So the language of nucleic acid present in the messenger RNA is translated into the language of protein. इथे ट्रान्सलेशन झालंय. ट्रान्सलेशन कशाचं झालंय? न्यूक्लिक ॲसिड्सचं ट्रान्सलेशन आपण अमायनो ॲसिडमध्ये करतो, म्हणून त्याची लँग्वेज बदलली. इथे लँग्वेज बदलली म्हणूनच याला ट्रान्सलेशन असं प्रोसेस म्हटलंय. So nucleic acid you have a 4-letter language, but proteins they have a 20-letter language. कारण proteins मध्ये 20 amino acids असतात. So how they are usually coded? 3 RNA bases forms a genetic code, and 3 RNA base code which we often called as genetic code specifies one amino acid. So this one amino acid is coded with 3 base sequence. So you have 4 different, means you have 4 nucleotide sequence. So this is your 4 nucleotides. So then you have this second 4 nucleotides and this is your first. So this is your first base, this is your second base and this is your third base. So how to read? You have to read this way. You are looking you are seeing the arrow diste tumhala. So this is your first letter U, okay. So this is your first letter U, which is this one. Then second U, this second U is your this U, and the third letter U is your this U. So U is coding the phenylalanine. Similarly, this 2 column and this row, okay. So if these 2 U's are remaining common in these 4 cases and this has been changing, then you will see this particular region, the third nucleotide is getting changed, okay. Then also they are specifying the same or different amino acids. In this way you will have, that is 4 to the power of 3 means 64. It will code 64 amino acids. It means that you can have 64 genetic codes. You can have 64. See why this 10 to the power of, see if it would have only 4 nucleotides, then it could not code for 20 amino acids. If it would have been, म्हणजेच 3 बेसेसच्या ऐवजी जर का एकच कोड असता तर एक कोड फक्त 4च अमायनो ॲसिड्सला कोड करता आलं असतं. U should have code for one particular amino acid, C would have coded for another, A would have coded for another and G would have coded for. पण आपल्याकडे किती आहेत? You have 20 amino acids, so it is not possible. Then 4 to the power of 2 जर का 2 यूज केले असतात, 2 च्या कॉम्बिनेशन म्हणजे UU, UC, UA, UG, ओके, किंवा CU, CA, CG, CC, तरी पण आपल्याला कॉम्बिनेशन हे फक्त 16चं झालं असतं, कारण 4 into 4 4 4s are 16, म्हणजे 16 अमायनो ॲसिड्सलाच कोड करता आलं असतं. म्हणूनच यू हॅव 4 raised to the power 3 means you can code for 64 amino acids and it is sufficient to code 20 amino acids. So you can code 20 amino acids with 4 raised to power 3, and this is how they are coded. Means the phenylalanine can have 2 codes, 2 genetic codes, UUU, UUC. Leucine can have 6 genetic codes, UUA, UUG, CUU, CUC, CUA, CUG. ठीक है. Methionine has only one code, AUG. Okay. Isoleucine has 3 codes. Then valine has 4 codes, serine has 4 codes, alanine 4, the threonine 4 codes likewise. Now you have the termination codons also, UAA, UAG and UGA. So these are your termination codons. So among the 64 codons, 3 codons are stop codons. You call it as the termination codons or the stop codons. Therefore, since one genetic code can go for, can specify many number of amino acids, this genetic code is called as a redundant for amino acids. Means 20 amino acids can be coded by 63 genetic codes. 63 genetic codes. Okay. So now how the translation takes place? Translation begins. So in certain cases On the messenger RNA, you have a leader which is not translated. This is what is called as a leader arm. So, in your next sem, you will see the biosynthesis of tryptophan. In biosynthesis of tryptophan, you will see that the tryptophan genes are at this location which are getting translated. But before that, in the biosynthesis, you have a leader arm which is usually not translated, but once translated, they can operate like a controlling mechanism, or they can regulate the biosynthesis of the tryptophan. So, messenger RNA, they can have a leader arm which cannot be translated. Then they have a reading frame which usually translates into a protein molecule, and you have a trailer which is non translated. This is also not translated with the stop codon. So, the start codon usually starts with the AUG, and the first amino acid is always the methionine. So, in prokaryotes you have N-formyl methionine, okay, and in eukaryotes you have the methionine. Later on, the 3 nucleotides are forming the genetic codes which specifies the amino acids, particular amino acids, and these particular amino acids makes the protein molecule. So, protein synthesis, how does it starts? Usually it is a process which takes place on the ribosome subunits. The ribosomes, you have 2 subunits, small subunit and the large subunit. The messenger RNA first binds with the small subunit, and then it forms a complex with the larger subunit. So, it forms a 3D groove, and this 3D, from on this 3D groove, usually the messenger RNA runs, or the messenger means the usually the ribosome subunits are static, means they sit on the messenger RNA, and messenger RNA is running 3 bases ahead. 3 bases ahead. Okay. म्हणजेच काय की रायबोसोम हा एकाच जागी थांबतोय आणि रायबोसोम मूव्ह होतो, सॉरी मेसेंजर आरएनए मूव्ह होतो, आणि मेसेंजर आरएनए जसा मूव्ह होतं तसे ते कोडॉन पुढे सरक, म्हणजे ते कोडॉन पुढे येतात आणि पुढे येऊन त्या पर्टिक्युलर टी आरएनए ती पर्टिक्युलर अमायनो ॲसिड घेऊन येते आणि ए साइटवर बाइंड होते. सो on the ribosome you have A site and P site. A site is the amino acid site and P site is the peptidyl site. So protein synthesis, that is usually the transfer RNA brings the amino acid which is attached to the 3' end. Where the amino acid is attached? Amino acid is attached at the 3' end. And which amino acid is usually attached here? It is attached according to the anticodon, because anticodon is forming a hydrogen bonding with the codon located on the messenger RNA. म्हणजेच मेसेंजर RNA चे 3 बेसेस तो रीड करतोय, त्याच्यावर तो tRNA आपले अमायनो ॲसिड घेऊन येतोय at the 3' end, and accordingly it forms a hydrogen bonding with codon and anticodon, and it places the that is tRNA along with the charged amino acid at the A site. Okay, so transfer RNA carries the amino acid to the ribosomes. So during tRNA charging, each tRNA picks up a specific amino acid. It picks up a specific amino acid from the internal, means in the periplasmic space, that is in the cytoplasm in the prokaryotic cell, that is intracellular. That particular amino acid is charged on the tRNA by tRNA synthetase. So, we will see in detail how the charging of amino acid is usually done. The ATP is coming into role. Okay. So, later on. So, usually the translation takes place in 3 steps: initiation, elongation and termination. Initiation, usually the 5' cap of messenger RNA, it binds to the ribosome. Then later on it starts the translation with the AUG codon, that is the start codon AUG, which binds methionine to the P site. The first amino acid is bound to the P site and A site is open and ready to receive the new tRNA. The second step is the elongation process. Elongation is nothing but addition of new amino acids with the formation of peptide bond. So, the codons are recognized and peptide bond formation takes place, and this after formation of the peptide bond, translocation is usually done. So, translocation means the ribosome is static but the messenger RNA moves along and amino acid tRNA shifts from A site to the P site. The termination is usually occurs at the stop codon, when the stop codon is usually reached. See UGA, or you have a sequence of 3 UAG, like that. So, when see here you have that is UGA as a stop codon. So, either UAA, UAG or UGA, whenever it reaches this particular codon, since this particular codon is not having any charged amino acid tRNA, the tRNA cannot come with the amino acid, and when it is not having any of the, means since it is not specifying any of the amino acid, usually a gap is created. So, when it is moving here, then the gap is created and the polypeptide is dissociated with the GTP molecule, with the hydrolysis of GTP molecule. So, this process or mechanism of translation we are going to see in detail later on. So, I hope you have understood this central dogma of molecular biology. If you have any doubt, you can ask now. Thank you.